Hello everyone. I welcome you all to this video lecture. We were studying about human reproduction and we have already seen up to the formation of embryo, great, the human embryo, that is up to the gastrula stage. Today in this video lecture we will be discussing about how the fetus development occurs, and then the formation of placenta, okay, the formation of extraembryonic membranes, organogenesis, and finally the parturition. Okay, so in this video lecture we are going to covering the rest of the human reproduction. Great. So in the previous lectures we have seen the formation of gastrula. We have seen how this different embryonic stages are formed. So we will do a slight recap and then we will proceed further in continuum. Great. So in previous lecture we have already seen the process of implantation. What I have drawn here, what we are seeing here is the blastula stage, which has been implanted, completely implanted inside the uterus, okay, in the uterine wall, and we have seen that the implantation preferably occurs in the fundic area of the uterus, right? Great. So this is the upper fundic area and the red part is endometrium. So in the endometrium area, this the blastula stage has come and implanted, okay, and see there is a red line, and this red line is the upper closing layer, so it is completely covered by the endometrial layer. Okay, great. We have also discussed in previous lecture that once this implantation starts, the cells of the trophoblast layer, they start forming this finger-like projections which degrade the uterine tissue and derive nutrition from it, and hence we called it fostorial, right? So it is kind of parasitic in nature. It degrades the uterine wall and derive nutrition from it, and this derived nutrition is utilized for the trophoblast as well as for the inner mass cells, both for the entire embryo, correct? And while doing that, while eating up or degrading the endometrial tissue, it made a pit for itself, and in this pit the embryo get dipped, and then it was covered by again the uterine tissue, completing the process of implantation. This we have already seen. So we know what chorionic villi is. Chorionic villi are these finger-like projections which are formed by the trophoblast cells, correct? Now, after blastula stage, once the implantation has occurred, we say that pregnancy is completed. The female is pregnant, okay? This is a hallmark of pregnancy. Now, after implantation, what happens is the inner cell mass, the cells of inner cell mass divide rapidly, okay, increase their number, and then they undergo a process of differentiation and then rearrangement. Okay, so differentiation and rearrangement. The rearrangement occurs because of the morphogenetic movement, right? And due to this, we have already seen that gastrulation occurs, right? So gastrulation occurs, the formation of gastrula, in which we saw that the inner cell mass cells differentiated into three types, that is the cells of ectoderm, okay, we can see here, ectoderm, mesoderm, and endoderm, and they moved in a particular fashion through two type of movement, okay, epiboly and emboli, correct? So this is all we have seen in the previous class, and as a result there is a formation of this three germ layers, ectoderm, mesoderm, and endoderm, and this is in a plate, right? So these three germ layers are formed, correct? Now these three germ layers are responsible for the formation of fetus, okay? The whole complete fetus or the child is formed from the cells of these three layers only, ecto, meso, and endoderm. The remaining cells, okay, so from this area cells, just a minute, from these cells, okay, Great. And the remaining cells will be utilized in the formation of other parts, that is placenta and the extraembryonic membrane. Okay, great. This we will see after a moment, how this ectoderm, mesoderm and endoderm leads to the formation of whole of the fetus or all the organs of a child. Okay, great. Now the formation of the 3 germ layers, when it initiates into the embryo, it leads to formation of a line-like structure, right? It's form a streak that is called primitive streak. Okay, that is called primitive streak. And actually this primitive streak further progresses and forms this entire germ layer. Okay, so a primitive streak is the initiation of the 2 type of movements, epiboli and emboli. Okay, and from there this streak grows and forms the 3 germ layers. Okay, this entire structure is formed. Now when these 3 germ layers are formed, the cells of these germ layers are differentiated cells. Okay, these has been differentiated from the cells of inner cell mass. Remember in the blastula, right? See here, the green one are the inner cell mass cells, and these cells are stem cells. These are the totipotent cells, as in these cells have total capacity. They have the capacity to form any organ or any tissue of a human being. Great. Now when the gastrulation occurs, these stem cells, these totipotent cells, differentiate into these 3 ecto, meso and endoderm, and when they differentiate, now they are determined to form a particular type of tissue. Okay, as in the ectoderm is determined to form certain type of tissues only, so does the mesoderm and the endoderm. Great. As in, if ectoderm is determined to form nervous tissue, the cells of ectoderm will form only and only the cells of nervous tissue. They are not going to, or they will not be capable to form any other cell, any other tissue. Okay, so this kind of determination is being done during the process of differentiation. Okay, now let us see which germ layer forms what kind of organs or tissues in our body. Okay, now this is called fate of the germ layers. Okay, fate matlab bhavishya, right? So fate of the germ layers, as in if we look at the ectoderm, ectoderm is going to form the nervous tissue. So the fate of ectoderm is always nervous tissue. This means that the cells of ectoderm will form the nerve cells or the accessory cells of the nervous tissue, such as Schwann cells, glial cells, astrocytes, and so on. Okay, great. When we talk of mesoderm, now mesoderm, the cells of mesoderm are determined to form the connective tissue and the urinogenital system. What does this mean? This means that the mesodermal cells or this mesodermal layer will be responsible to form all type of connective tissue, such as muscle cells, both skeletal and smooth, as well as the cardiac. Okay, it will be responsible for the formation of all the bones, cartilage, blood, lymph, and so on. So all the connective tissue, okay, such as areolar tissue, adipose tissue, all these tissues, as well as the urinogenital system, testes, ovary, right, ureter, these all will be formed by the mesodermal cells. Great. Finally, if we look at the endoderm, now the endoderm is going to form the epithelial lining. Lining is the innermost covering, okay, innermost layer. So the lining of the internal or visceral organs, such as respiratory system, digestive system, and so on. So if you see the nervous tissue, is formed by ectoderm. The connective tissue is formed by the mesoderm. What about epithelial tissue? The epithelial tissue can be formed by all of these three germ layers. It can be formed by ectoderm, mesoderm, and endoderm. Okay. In any organism, the epithelial tissues are formed by all these three. Okay. So different epithelial tissue can be of different origin. They can either be ectodermal in origin, or can be mesodermal in origin, or can be endodermal in origin. Okay. So do remember that epithelial tissue is the only tissue that has origin from all the three germinal layers. Okay. Now these are called germinal layers. Ectoderm, mesoderm, and endoderm are called germinal layers. Okay. So epithelial tissue can be developed from any of the germinal layers, whereas nervous tissue only from ectoderm, connective from mesoderm. याद रखिएगा इस चीज को. Great. And only these three germ layers, the cells of these three germ layers, are responsible for the formation of fetus. Okay. One more thing. Now when this gastrulation occurs, the cells migrate from this end. Okay. So the movement of the cells occurs from this end inside this layer to form the primitive streak, and later on this entire germinal layers. Okay. So this entire layer is formed because of the movement of the cells, and there is a pore-like structure through which the cells move in. Now this pore is called blastopore. This is called blastopore. Okay, and the side of the blastopore is called vegetal pole. This is called vegetal pole. On the other side, this part is called animal pole. Okay, so this is animal pole, and towards the blastopore is the vegetal pole. The movement, the morphogenetic movements during the gastrulation occurs through the blastopore or through the vegetal pole. Okay. Now, if you look at the genesis of fetus or the embryology, what we find is the head is formed from the animal pole side, whereas the tail or the legs is on the vegetal pole side. Okay. So what we will see is this entire thing, this three germinal layers. Here it will develop the head and then the body, and this will develop into the tail part of the fetus. Okay. Now apart from this, these cells, these layers, can you see these layers and these layers? These will develop into placenta and extra embryonic membranes. So when we look at this entire embryonal structure, later this embryonal structure is going to develop into three structures, right? What are these three structures? Number one is the fetus itself, which is the most important thing here. Fetus, and then is the, okay, so this entire structure, the fetal structure, the embryo gastrula is going to develop later into these three structures: fetus, placenta, and extra embryonic membranes, right? Out of which the fetus is going to develop from the embryonal axis, okay, from the embryonal axis, which is made up of the germinal layers, three germinal layers. Placenta is mainly going to form from the chorionic villi and maternal tissue, okay, while the extra embryonic membrane is going to be formed from the part of embryo, okay, from the parts of embryo. Great. So this entire structure is now going to get segregated into three parts, that is fetus, placenta, and extra embryonic membranes. How do they look? Let us have a figure here. Now this figure is from NCERT, as most of you know. So if we look at this figure, can you see? Here was the implantation, right? Earlier, here was the implantation with this finger-like projections here, embryonic villi, and here was the formation of those embryonal axis from which if we see from this embryonal axis, here the development of head occurred, and then the body, and then the tail. Remember? If it is difficult to see, now see in green color. This was correct. This was the implanted embryo, blastula, which has chorionic villi, and then during gastrulation there was formation of this embryonal axis, 3 germinal layers, and from the animal pole formation of head, and then body, and then tail part of the fetus, right? And this is what you see here. See, now it is a NCERT figure showing the embryo, right? Can you see? This is the head of the embryo, this is the body, and this is the posterior part. It is still having the tail, which will be later replaced by the legs, right? And this embryo, growing embryo, is going to require a lot of nutrition, and this nutrition will be provided from the mother through placenta, okay? But from placenta, it will reach up to the embryo via this particular tissue called umbilical cord. Okay, so basically umbilical cord is a rope-like structure, okay, which is consist of different blood vessels, okay, blood vessels of the fetus, okay, the blood vessels through which fetus is directly connected with the placenta. Great. So this is how it looks. So can you see now here this implantation has occurred in the fundic region, and from there it is growing the embryo, and there is placenta, and the outer covering of that embryo has now developed into extra embryonic membranes. Okay, we will talk in a while about this extra embryonic membranes. Great. Now, so we now know that there will be 3 structures: fetus, placenta, and extra embryonic membrane. So one by one we are going to see them. Well, as an overhaul, see this figure. Now in this figure you can see again this is the fetus, and it is connected to this placenta via a rope-like structure called umbilical cord. Okay, and this placenta is embedded into the uterine wall. Okay, if we zoom in and try to study the placenta, what we find is, see, those chorionic villi, the chorionic villi of the embryonic stage, as the fetus develops, the chorionic villi also develops, okay, and now it forms a tree-like structure called chorionic villi or chorionic tree. Okay, now this chorionic tree-like structure is highly vascular and branched, okay, and it is made up of the blood vessels from the fetus, right? So it is made up of the blood vessels from the fetus. याद रखिएगा बेटा. Okay, chorionic villi is a fetal structure. ये maternal structure नहीं है. देखो जब placenta बनेगा बेटा, placenta is made up of both fetal tissue and the maternal tissue. Okay, so there is fetal tissue as well as the maternal tissue. They both contribute to form this disc-like structure called placenta, which is utilized mainly for the exchange of materials between the fetus or growing fetus and the mother or maternal tissue. Mainly, placenta is responsible for the transportation of O2, nutrients, okay, other useful things, antibodies like IgA to the fetus from mother. At the same time, it is responsible for withdrawing the waste product such as CO2 and nitrogenous waste such as urea from the fetus into the maternal blood or maternal tissue. Okay, तो ये जो प्लासेंटा है, it is like the transport hub, okay, between the mother and the growing fetus. जिससे क्या होता है, जो fetus है, it is taking the useful things from the mother. At the same time, it is giving out all the harmful things to the mother. तो मदर इसको सारी यूजफुल चीजें दे रही है और फीटस अपना सारा जो भी वेस्ट प्रोडक्ट है वो मदर को दे रहा है. दिस इज द मेन फंक्शन ऑफ प्लेसेंटा. एंड देयरफॉर दिस प्लेसेंटा हैज टू बी मेड अप ऑफ बोथ मैटरनल एंड द फीटल टिश्यू. क्यों? क्योंकि यहाँ पे द प्लेसेंटा मेक श्योर दैट देयर इज नो डायरेक्ट मिक्सिंग ऑफ द फीटल एंड द मैटरनल ब्लड. याद रखेंगे आप लोग, इन प्लेसेंटा डायरेक्ट mixing of maternal and fetal blood does not occur. ऐसा इसलिए नहीं होता है क्योंकि it is not necessary that the maternal blood and the fetal blood are of same type. Okay, हो सकता है कि mother is A positive and the child is B positive. That is possible. Okay, और ऐसा होता है. अगर direct mixing of blood होगा तो क्या होगा? The fetal RBCs will clump because of incompatibility between the two blood groups and the fetus will die. Abort हो जाएगी प्रेगनेंसी. सो इस चीज से बचने के लिए कभी भी मैटरनल और फीटल ब्लड की मिक्सिंग नहीं होती है. द प्लासेंटा मेक श्योर कि दोनों के ब्लड की मिक्सिंग नहीं हो. ठीक है, तो प्लासेंटा का कुछ ऐसा स्ट्रक्चर ही है कि डायरेक्ट मिक्सिंग नहीं होती है. इसीलिए क्या है, ये दोनों टिश्यूज का बना हुआ है. जो कोरियोनिक विलाई है इसमें, इट इज फिंगर लाइक प्रोजेक्शन विच इज इंटरडिजिटेटिंग इनटू द यूटेराइन वॉल. इंटरडिजिटेटिंग का मतलब होता है उंगली जैसा स्ट्रक्चर है जो कि अंदर धंसा हुआ है. ओके, इट इज पेनिट्रेटिंग इनटू द यूटेराइन वॉल, इनटू द मैटरनल टिश्यू लाइक फिंगर्स. ओके, ब्रांच्ड फिंगर्स, जैसे कि ट्री जैसा स्ट्रक्चर, उसके ब्रांचेस हो. ओके, और कोरियोनिक विलाई के अंदर होते हैं बहुत सारे ब्लड वेसल्स, और ये ब्लड वेसल्स किसके होते हैं? फीटस के. पूरा कोरियोनिक विलाई किसका बना हुआ है? फीटल टिश्यू का. ग्रेट. नाउ अराउंड द फीटल टिश्यू इज द यूटेराइन टिश्यू, और यूटेराइन टिश्यू किसका है? मदर का. ओके, नाउ दिस यूटेराइन टिश्यू इज एक्चुअली द पार्ट ऑफ एंडोमेट्रियम which is highly vascularized. Now around each chorionic villi there is a space, and this space is called intervillous space. This is called intervillous space. दो कोरियोनिक विलाई के बीच के स्पेस को क्या बोलेंगे? इंटरविलस स्पेस. करेक्ट. और ये जो इंटरविलस स्पेस है, इस स्पेस में चारों तरफ से मैटरनल ब्लड भरा होता है. सो दिस एंटायर कोरियोनिक विलाई इज एक्चुअली submerged or is dipped into the maternal blood. समझ में आ रहा है? तो कुछ आप ऐसा समझिए कि this is the intervillous space, जिसमें कि एक विलस है और इसके अंदर यहां पर एक कोरियोनिक विलाई है. ट्री जैसा स्ट्रक्चर है ये. ओके, जो कि ऐसा जा रहा है, और इसके अंदर फिर आपके बहुत सारे ब्लड वेसल्स हैं. ये सब फीटस के ब्लड वेसल्स हैं, पर ये इसके अंदर हैं, इसके बाहर नहीं है. और ये सब मिलकर के फाइनली यहां से निकल जाते हैं. और ये जो पूरा का पूरा कोरियोनिक विलाई वाला स्ट्रक्चर है, इट इज बीइंग डिप्ड इनटू द मैटरनल ब्लड. सो ये जो पिंक वाला है उसके अंदर फीटल ब्लड है और कोरियोनिक विलाई के बाहर मैटरनल ब्लड है. सो इसकी वजह से क्या हो रहा है, जो मैटरनल ब्लड है और जो फीटल ब्लड है वो कभी भी आपस में मिक्स नहीं हो रहा है. कैन यू सी दिस एरिया? दिस इज ऑल मैटरनल ब्लड. द रेड पार्ट इज ऑल मैटरनल ब्लड. Okay, whereas the fetal blood is only in these red and blue blood vessels, that is artery and veins of the fetus. So there is no mixing, never. Great. Now the blood which is present in this intervillous space is carried by these spiral veins and arteries, which are called uterine artery and vein. These are spiral in nature, therefore we also called spiral blood vessels, okay, and these finally are attached to the larger artery and veins of the maternal tissue. Okay, so these all are attached to the larger maternal artery and veins, which are actually the uterine artery and veins. Great. So this entire structure, if you see this part where chorionic villi is there, the maternal tissue part, okay, from this up to this, the maternal tissue part, which is participating in the formation of placenta, is called decidua basalis. This is called decidua basalis. Why we call it so? We call it so because it is deciduous in nature. अब deciduous तो आप लोग जानते हैं, deciduous forest, ऐसा फॉरेस्ट जिसमें कि पत्ते गिर जाते हैं. वैसे ही आफ्टर प्रेगनेंसी जब पार्ट्यूरेशन होता है तो ये ऐसा प्लासेंटा का लेयर है जो कि फीटस के साथ टूट करके निकल जाता है. ओके, सो इट इज deciduous in nature. So it is being shed off after the completion of pregnancy. Great, and that's why this layer is called decidua basalis, and this decidua basalis is actually the maternal tissue or maternal part of the placenta. It includes the intervillous spaces and the spiral blood vessels of the uterine artery and vein. Okay, great. Now, and this whole structure, if you see this whole structure of placenta, can you see it is a disc shape? Okay, so it is discoid in nature. ठीक है, ग्रेट. अब देखिए हम लोगों को एक जो सबसे इंपॉर्टेंट चीज ध्यान रखनी है, दैट इज द फंक्शन ऑफ प्लासेंटा. ओके, सो आई एम गोइंग टू राइट इट हियर. Please make a note and do remember them, that the function of placenta is number 1, exchange of materials, okay, जिसमें से कि from mother to fetus it gives O2, it gives nutrients, okay, it gives vitamins, it gives minerals, it gives water, it gives IgA antibodies. Okay, these are the only antibodies, sorry, not IgA, I'm extremely sorry on this, IgG. Okay, these are the only antibodies that can cross the placenta, okay, and reach up to the fetus from the maternal blood. ठीक है, तो बार बार ये सवाल आपसे पूछा भी जाता है that which antibody can cross placenta. The answer is IgG. Great. Apart from that, if we look at the exchange of materials from mother to fetus, ये सारी चीजें जाती हैं. From fetus to mother, from fetus to mother किसका exchange होगा? The exchange of CO2 waste product, CO2, as well as nitrogenous waste, that is mainly यहाँ पे क्या होगा? यूरिया. We are eutelic animals, that's why our main or chief nitrogenous waste is urea. Great. Now one thing to remember here again is, apart from these things, certain other things such as alcohol or other drugs can also cross through the placenta. Okay, so if a pregnant woman is intaking alcohol or taking drugs like aspirin, they can cross the placenta and go to fetus, and this has a very bad effect on the fetus. If a pregnant woman is taking aspirin, which is a painkiller, okay, this aspirin can lead to a disease called phocomelia in the fetus, in which the development of limbs is retarded or is affected in the fetus. So as a result, if a female is taking a lot of aspirin during her pregnancy, her child could have deformed uh, limbs. Okay, so he or she may have deformed hands and legs, and the disease is called phocomelia. Okay, or if the female is alcoholic, or she uses a lot of tobacco, or she has any other she is abusing any other drug, it could have detrimental physical and mental effect on the growing fetus. Okay, great. apart from this Apart from this, what we see here is the placenta also ensures non-mixing, right? Non-mixing of maternal and fetal blood. Okay, so if we consider the functions of placenta, what we find is the main function of placenta is the exchange of materials, okay, such as exchange of O2, CO2, nutrients, vitamins, minerals, water, okay, nitrogenous waste, or even the exchange of some other drugs or alcohols that occurs through placenta. We also saw that through placenta, the smallest antibody, that is IgG, can cross and go to the fetus. As a result, the mother provides passive immunity against certain diseases such as chickenpox, measles, polio to the growing embryo. She may give if she already have those antibodies. If she don't, then there is no chance. Okay, great. What else placenta does? Okay, another aspect of placenta is it acts as endocrine gland. Okay, so placenta acts as endocrine gland and it secretes a lot of hormones. What are these? Number one, it secretes HCG. The full form is human chorionic gonadotropin. Okay, human chorionic gonadotropin, and HCG is a very, very important hormone for the pregnancy. Why? It is responsible for maintenance of corpus luteum. Okay, corpus luteum for up to 2 months. Okay, so HCG is being secreted by the chorionic villi, and as soon implantation is done, immediately after that it starts to secrete HCG, and this HCG reaches up to the corpus luteum inside the ovary of the female or the mother. Okay, and it makes sure that the corpus luteum do not degenerate in next 9 days. Rather, it stands there for the for next 2 months, that is up to the end of 2 months. And by doing so, this HCG makes sure that corpus luteum keeps on secreting progesterone, okay, which is responsible for the maintenance of uterine wall and hence the implantation. Okay, so if there will be no progesterone, the endometrial lining will not be maintained, and if there is no endometrial lining, obviously there will be no implanted embryo, so no pregnancy, right? So HCG makes sure that it maintains the corpus luteum, which in result keep producing progesterone, which is responsible for maintenance of the endometrial lining and hence the pregnancy. Okay, great. What else? So this is one hormone that it secretes, placenta. The second hormone is called HPL. Okay, HPL, the full form is human placental lactogen. Okay, now HPL, human placental lactogen, is responsible for development of mammary gland. Okay, development of mammary glands. Okay, so it is actually preparing the female's body for the motherhood. What is third? If we are talking of the third hormone, it secretes estrogen. Okay, and estrogen is responsible, or it is required for the uterine growth as well as for the development of mammary gland. Okay, so estrogen, again, it is responsible for uterine as well as the mammary gland growth. The third hormone that it starts to produce is progesterone. Okay, is progesterone. So see, the progesterone is initially secreted by corpus luteum. Up to the second month, it is mainly secreted by the corpus luteum, but after 2 months, corpus luteum is going to degenerate and become corpus albicans, right? After that, who will produce the progesterone? The placenta itself. Okay, so the placenta itself keep on producing the progesterone, which is responsible for the maintenance of pregnancy. Maintenance of pregnancy. Apart from this, what we see is the placenta is also responsible for the secretion of relaxin. Okay, so in later phase of pregnancy, relaxin is secreted by the placenta, which is responsible for parturition. Okay, for parturition, that is childbirth. Great. It initiates the process. Okay, it helps in parturition. Great. Apart from this, what we find is during pregnancy in a female, the other hormones such as cortisol and thyroxine levels become high. So they become higher multiple folds, the cortisol and thyroxine. Why it is so? Because cortisol, remember, cortisol is responsible for carbohydrate metabolism, and it was secreted by the adrenal cortex, right? The glucocorticoids. It is the chief glucocorticoid, cortisol, and it is responsible for increasing the carbohydrate metabolism into the female body, which is required for more and more energy production. Now remember, the pregnant women, her energy demands are higher because she has to take care of the energy needs of herself as well as the growing fetus inside her, and therefore the cortisol levels are increased multiple fold. Similarly, thyroxine, it increases the basal metabolic rate, which is again required for better oxygen supply and other nutrient supply to the fetus. So both this cortisol and thyroxine levels are increased multiple fold in a pregnant woman as to increase her, or sorry, as to prepare her body for the pregnancy. Great. So what we have seen so far is the placenta has very, very distinct functions. Apart from controlling the exchange of material, it is also acting as an endocrine gland, and it is secreting a lot of hormones which is responsible, or which is required for the pregnancy, for a healthy pregnancy. Okay, great. So this was placenta. If we move to the next part, that is extraembryonic membrane, what we find is, let us look at this figure again. What we find is the embryo is surrounded by few extraembryonic membranes, few membranes which are around this fetus or the embryo, growing embryo, and hence we called extra embryonic membranes. What are these? The first and outermost is chorion. Okay, the first and outermost is chorion. Inner to the chorion, what we find is amnion. Okay, and then we find two more, yolk sac. Okay, sorry, one more, yolk sac. So look at this figure. We are zooming in. What we find here is this outer part. Can you see here this outer layer? This outer layer, this outer membrane is called chorion. This is chorion, and it is mainly made up of the ectoderm. Okay, well, if we look at this chorion, it is made up of two layers. Okay, so it is bilayered, and of these two layers, the outer layer is ectoderm and the inner layer is of mesoderm. Okay, so let me write it here. If we are talking of chorion, which is the outermost extra embryonic membrane, it is made up of ectoderm and mesoderm. If we are talking of amnion, it is made up of a single layer of ectodermal cells. And if we are talking of yolk sac, which is also known as allantois, it is made up of mesoderm and endoderm. Okay, so please remember that there are these three extra embryonic membranes. Okay, the outer one is chorion. The outer one is chorion, which is made up of two layers. One is ectoderm and the outer one, and the inner one is of mesodermal cells. Okay, and it is the chorion is the outermost post extra embryonic membrane which protects the embryo inside the uterus. Okay, now on the inner side of the chorion there is another extra embryonic membrane called amnion. Okay, and this amnion is single layered membrane which is made up of flat ectodermal cells, and inside the amnion layer a fluid is filled. Okay, so can you see this green one? This green one is amniotic cavity. This is amniotic cavity because it is a space which is enclosed by the amnion membrane. Okay, and in this amniotic cavity there is a fluid filled. Okay, this fluid is called amniotic fluid. Okay, this is called amniotic fluid, and the embryo actually keeps swimming in this amniotic fluid. Okay, so this amniotic fluid is present inside this amniotic cavity in which the embryo is actually floating or it is swimming, and therefore what we find is that the embryo development occurs inside the water. Okay, remember, even in humans the embryonic development occurs in water. Which water? The amniotic fluid, which is enclosed by the amniotic membrane or which is present in the amniotic cavity. Right? Now, apart from this amnion, what we find here is there is one extra membrane, extra embryonic membrane, that is called yolk sac or allantois. And this allantois is made up of mesoderm and endoderm. It is yolk sac. In human beings it is reduced, highly reduced, but in case of other animals like birds, reptilia, fishes, it is developed and it actually possesses the yolk, which is the nutritive substance or food for the growing embryo. Okay, so in all these animals in which the amount of yolk in the egg is more, the yolk sac is larger and it contains the yolk. Hence it is called yolk sac. Great. And since we are talking of this, let me tell you that on this basis, the eggs are of different types, such as alecithal, the eggs which do not have yolk, called alecithal. Okay, microlecithal, mesolecithal, and megalecithal. So there are 4 types of eggs on the basis of the amount of yolk. If there is no yolk, alecithal. If there is very small amount of yolk, microlecithal. If there is moderate amount of yolk, mesolecithal. And if there is large amount of yolk, we call it megalecithal eggs. Such as megalecithal eggs are the eggs of birds, okay, or reptiles. These are megalecithal eggs because they have large amount of yolk in them. Great. Anyway, that is not the part of the syllabus, so we are not covering it here. And the eggs are of various types. The other type of egg is a cladoic and non-cladoic egg. Cladoic egg is the egg having the hard outer shell, as you see in the chicken egg. Okay, there is a white shell, hard shell, which is made up of calcium carbonate. So such an egg is called cladoic egg. And the egg which do not have hard shell around them are called non-cladoic egg. Say for example the eggs of fishes or the eggs of human beings, that is non-cladoic egg. Okay, eggs are of different types. They can be classified into different groups on different bases. Right, all you need to remember is cladoic, non-cladoic, and alecithal, mesolecithal, microlecithal, and megalecithal. That's it, not more than that. Great. So what we have seen so far in this video is placenta and post extraembryonic membranes. So there are 3 extraembryonic membranes: chorion, amnion, and allantois or yolk sac. Correct? We have seen what they are made up of. Now we have also seen that the function of yolk sac or allantois is to contain the yolk, which is the food for the growing embryo. Okay, chorion has protective function, and amnion, it contains the amniotic fluid, and the function of amniotic fluid is it is going to provide a shock absorbance to the growing embryo or the fetus. Great. Very well. Apart from this, the amniotic fluid also serves another purpose for us. Using this amniotic fluid, we can do a particular test called amniocentesis. Okay, amniocentesis. What is it? Amniocentesis is a procedure in which we carefully take small sample of this amniotic fluid from the developing embryo or from this amniotic cavity. Okay, how do we take it? So under the ultrasound, when we can see where the baby is, we carefully, under the ultrasound observation, we prick a needle into the belly of the mother and take small sample of this amniotic fluid. This amniotic fluid contains some of the cells of the fetus, growing fetus. By analyzing those cells of the fetus, we can actually detect if the fetus has any genetic disease or not. Okay, so the main function of amniocentesis is the diagnosis of any genetic or hereditary or metabolic diseases before the childbirth. Okay, so it is usually done around 3 months. Great. So these are the functions of the extraembryonic membranes. Now let us focus on the fetal development. Fetal development. So if we study the fetal development, what we find is at different time points we find different structures into the human embryo. Okay, and the first organ that develops, the first organ developed is heart. Please बेटा याद रखिएगा, क्वेश्चन पूछा जाता है कि which organ is first developed in an embryo, and the answer is heart. कब बन जाता है? After first month. Okay, so once this first month is completed, by the end of first month, heart is almost formed. Okay, and after 1 month you can listen the heartbeat. Okay, so after 1 month the heartbeat of the fetus is audible with the help of that stethoscope. Okay, and this heartbeat is actually a measure of fetal health. Okay, so if the heartbeat is good, if the doctor can listen the heartbeat, the development of fetus is growing good. What they actually count as the heartbeat, the number of heartbeat. Now in this case, the fetal heart rate, okay, so the fetal heart rate is about 140 beats per minute, okay, to almost 180 beats per minute. So the fetal heart rate is really very, very high. Okay, and by counting this heart rate, by listening the sound of the heartbeat, the doctor can judge the health of the growing fetus. Okay, in the womb or uterus, or pregnant woman uterus is called womb. Okay, WOMB. So the fetal health is mainly judged on the basis of fetal heart rate and the sound for about 1 to 3 months of the pregnancy, even after that. Great. Now, as the pregnancy goes on, what we find is by the end of second month, okay, by the end of second month, the limbs are formed. Limbs and digits are formed. Now, what we mean by limbs and digits? Limbs are your hands and legs, and digits are the fingers. Okay, the fingers and the toes are the digits. So by the end of second month, what we find in a growing fetus is it has a heart and it has limbs. It has hands and legs, though they are very small, okay, but they have hands and legs with the small little fingers and the toes. So these all are formed by the end of second month. Further, what we find is by the end of third month, okay, third month as in the first trimester or 12 weeks. Remember, mostly the pregnancy is counted in weeks, not in months. Okay, so first week, second week, third, fourth, fifth, and so on. So mainly a normal healthy pregnancy is for around 40 weeks. Okay, for around 40 weeks, and the pregnancy is mainly studied in weeks. Great. Now we already know, I know that most of you will be knowing already, that the time frame for human pregnancy, a normal healthy pregnancy, is around 9 months, and this time is called gestation period. This is called gestation period. So the gestation period for humans is 9 months. We have already discussed all this in chapter 1. Now these 9 months are divided into 3 parts. Okay, first trimester, second trimester, and third trimester. Trimester because each part is consisted of 3 months, right? So these 9 months are divided into 3 parts of 3 months each, and they are called trimester, first, second and third trimester. So by the end of third month, that is by the completion of first trimester or 12 weeks, what we find is the embryo has developed considerably and all vital organs are formed. So if we are talking of heart, kidney, liver, okay, all spleen, all these organs have already been formed by the end of 3 months. Okay, now the process of formation of organs from the tissue is called organogenesis. It is called organogenesis. So what we find is by the end of third month, the organogenesis is almost completed. Okay, so by the end of first trimester, the organogenesis is complete. All the major organs have been formed. Now, further, when we track the growth of the fetus, we find that by end of fifth month or in fifth month, okay, by fifth month, what we find is the hairs on head has grown. Okay, so in fifth month we start to see hairs on the head of the fetus, as well as slight movement of limbs, slight movement. Okay, so by fifth month we start to experience slight movements, mainly the movements of limbs. Great. What we see by end of 6th month, by end of 6th month, that is second trimester, what we see is the hairs have grown all through the body. Okay, by 5th month it was only on the head, but now by 6 months body hairs can be seen all across the body, which is the mammalian character, right? Body hairs is a mammalian character, and now this character has appeared by the end of 6th month. Okay, body hairs, as well as what we see is the two eyelids. We have two eyelids, upper and lower eyelid. The both eyelids have separated. Okay, this means that now the baby can blink her eyes, his or her, the fetus can blink his or her eyes. The eyelids are separate now. So up to 6 months they are kind of blind. They cannot see, they cannot separate their eyelids because they are fused. Okay, only by the end of 6 months they are separate, and now they'll start opening or blinking of their eyes. However, inside the womb it is pitch dark, so it's of no use as of now. Okay, what else we see? We also find that the eyelashes have grown by end of 6th. So by the end of 6 months you find that the growing fetus can open their eyes, they have eyelashes, they have body hairs, there is more movement of limbs. Okay, from the 7th month onwards the female experiences, or she can feel a lot of the movements. Okay, movement of both hand and feet. Great. So these are some of the milestones of the fetal development in the chronological order through the gestation period. Now after 6th month, 7th month, 8th month, and slowly the body of the fetus grows. Okay, and as the 9th month is completed, end of 9th month, that is third trimester, the pregnancy is complete, the fetus is ready to come out of the womb. Okay, and by the end of 9th month the fetus itself, the placenta and the female body, they act in synergy. they act with each other to initiate the process of parturition. Okay, parturition, that is childbirth. And this initiates with the release of relaxin by the placenta. Okay, so the placenta releases relaxin, and then there will be release of oxytocin from the from the pituitary, okay, posterior lobe of pituitary, and that will initiate the process of parturition. As a result, the childbirth, the process of childbirth will occur, and finally the child will be, or the fetus will be expelled out. Okay, so the fetus will be expelled out of the uterus through the birth canal, and finally the childbirth will take place. Great, we will see it. So this is all for this lecture. In the next lecture, we will be talking of the parturition and the whole mechanism of parturition. Lactation, we will be talking about the other conditions such as the twins, mono and dizygotic twins, and with that this chapter will finish. So there will be one more lecture in this chapter, and the third chapter, that is human reproduction, will be finished. Great. So we will meet in the next lecture. Till then, take very good care of yourself. Thank you very much for being with me.